All right, welcome. We've got some pretty serious notes to take today. We are going to look at chromosomes and M phase of the cell cycle. So without further ado, let's get to it. So these are titled 8.4, Chromosomes and M phase. M phase, and that's the division of the portion of the cell cycle where first our nucleus is dividing and then the cell itself divides. So we'll get to that in due time. Let's start off though with DNA. So DNA and the proteins that help to organize it. So DNA is not all by itself. It's not just these strands. There's actually quite a few proteins involved, and we'll talk more about them in a couple chapters when we come back to DNA. But the DNA and its proteins are found in strands. So think of like a piece of yarn, and they're called chromosomes. Eukaryotes, so these are plants and animals like ourselves, we have various numbers of, and not unique numbers of, chromosomes in each of our cells. So what I mean by various numbers and unique amounts of, so for example, fruit flies, have 8 chromosomes. Humans, perhaps you've heard, have 46 chromosomes. Again, perhaps you're aware of that. But there you can see, depending on what type of eukaryote we're looking at, they have various numbers. Fruit flies have 8, humans have 46. Now again, these numbers are not unique to the species. So they're not unique. Humans are not the only organism that has 46 chromosomes. The sable antelope over in the plains of Africa, as well as others, so et al., Latin for and others, also have 46 chromosomes. So 46 is not unique to humans. Lots of species, or quite a few of them at least, have 46 chromosomes. So we have various numbers, but they're also not unique. Don't think that the more chromosomes you have makes you a more complicated species either. I think we usually have pretty high opinions of ourselves, Homo sapiens. Sapiens means intelligent, so we've named ourselves intelligent. Go figure. Okay, yes, we are probably quite a bit more sophisticated than a fruit fly. But don't think that that has anything to do with the number of chromosomes that we have. Red king crabs have 206 chromosomes. So they have a lot more than we do. I wouldn't necessarily say that they're any more sophisticated than ourselves. And then the most I've ever heard of is there's a specific type of fern that with a plant. So plants are also eukaryotes. They also have individual number of chromosomes, but there's a specific type of fern that has as many as 1,260 chromosomes. Okay, on a double exclamation point. So yeah, the more chromosomes you have doesn't necessarily mean a thing. So that's eukaryotes. We'll mention prokaryotes as well. There's a lot more of them than us, so we shouldn't just ignore them. Prokaryotes tend to have just a single chromosome. And unlike ours, which are, again, think of a piece of yarn, a long piece of yarn, prokaryotes tend to be circular, more like a rubber band, really. So prokaryotes have one single circular chromosome. I've got some more definitions, more terms that go with chromosomes, and it'll be important to know these terms as we continue with our look at the events of M phase. So for much of interphase, which remember, we talked about this a couple days ago, interphase represents the majority of a cell's life. Cells are not dividing all that often. At least 3/4 of their life, I would say, is spent in interphase. And for a lot of cells, a lot longer than 3/4 of their life is spent in interphase. So the majority of a cell's life is spent during interphase. And during this time, so during interphase, for most of a cell's life, chromosomes are loose and unraveled. They're uncoiled. They're not very well organized. And this form is called chromatin with an N. I always think if you could take the nucleus, remember with eukaryotes, our DNA is stored up within a nucleus. If we could take that nucleus and cut it in half, you'd have what looks like a bowl full of spaghetti. Where the chromatin is the spaghetti. The bowl then is the nuclear envelope, the shell of the nucleus. But chromatin looks like spaghetti, meaning it's going to be a big mess. You can't tell one chromosome from another. You can't tell where one ends and one begins. It just looks like a big knot of spaghetti, and that's probably what your DNA looks like most of the time. It's in its chromatin form. It's loose. It's unraveled. It's not organized. You can't tell individual strands apart. It all just looks like noodles. It just looks like spaghetti. And that is chromatin. While it's in that chromatin form, during S phase, so during S phase, and the S stands for synthesis, we're making DNA. So during S phase, each of those DNA strands, again, you have 46 strands in each of your cells. So each of those 46 DNA strands is duplicated. And S phase stands for synthesis. What are we synthesizing? DNA. So those duplicated chromosomes are held together at what we call the centromere. The centromere. Kind of as the word states, center. Most centromeres are found towards the center of the middle of the chromosome, and that ends up why most of the time chromosomes look like X's to us. They're kind of held together in the middle. So it's not until we get to the start of mitosis does that chromatin, that bowl full of spaghetti, condense. It coils very tightly, and it condenses into what we call chromatids. So there's another term that refers to DNA, but it has ever so slightly different meaning, which is why we're going over all this before we get started. So chromatids The chromatids are a more organized and tightly coiled form of chromosome. And it's only in their chromatid form where individual chromosomes are visible. So sticking with that bowl full of spaghetti, we can see the DNA, we can see the spaghetti, but we can't see individual chromosomes, kind of like we can't see individual noodles. It all just looks like one big old mess. Well, what if we go backwards with our spaghetti? You know, spaghetti started off as those straight strands that you could easily identify, and we cooked it, we softened it up, and it turned into that mess. What if you could go backwards and go from that soft mess back into those straight strands that you can individualize? Well, that's kind of what happens when chromatin goes back to chromatid. It almost goes backwards. They become those straight, visible strands of DNA that now we can see and identify one chromosome from another. So it's only in the chromatid form that we're able to see individual strands of DNA. And here's the kicker. So by the time we can see those chromosomes, S phase has already occurred. The chromosomes have already been duplicated. So normally when we see chromosomes, when we're able to see them individually, they look like X's. They've already been duplicated. So when we usually think of chromosomes, we usually think of an X shape. But what I'm trying to get through to you is that for most of a cell's life, it's not an X shape. An X shape is a duplicated chromosome, and that only happens during S phase. And you go through G2, and those X's get pulled apart. And so for most of a cell's life, or much of a cell's life at least, those chromosomes are not X-shaped. They look more like an I, a capital I, or a lowercase l. But we just can't see them. They're loose, they're unraveled, they're in the chromatin form. All right, so we've thrown out a few terms here. We got a few more to go. Let me go ahead and flip the page here. All right, I'm going to give ourselves a diagram here. I think a picture's worth 1,000 words. So, well, let's do that. Huh? So we'll call this a cell. Here's the nucleus, and here's what DNA generally looks like. You can't really see much. You can see it, but you can't really see much. Well, guess what? This is chromatin. This is chromatin. And when it's in its chromatin state, the individual chromosomes are not visible. How many are there? I don't know. Could be a lot, could be a few. Maybe we're looking at a fruit fly cell. Maybe we're looking at a human cell. Maybe we're looking at a king crab cell. It's tough to tell. That DNA is loose. It's unraveled. It looks like a bowl full of spaghetti. Versus this nice organized nucleus with those individual chromosomes that we can actually see. This is DNA in its chromatid form. Chromatid form. Now, so now, by this point, by the time we see them in their chromatid form, by the time they've coiled up and condensed, they're now individual chromosomes. We can see them. They've already duplicated. That's why they look like X's. We've got two strands here. We've got one on the left here and one on the right. That is a duplicated chromosome held together in the middle at the centromere. And so that's why they look like an X to us. The only time we can see them is when they coil up into chromatid, and that happens only just before mitosis, or just in the pre-stages of mitosis, the beginning stages of mitosis, and by that point, they are duplicated. So that's why we think of an X when we think of chromosomes. Again, I'm going to continue, add a few more diagrams here. Again, I think pictures are worth 1,000 words, so let's give you 1,000 pictures, huh? Not much of an artist, but what I'm attempting to show you here, these are chromosomes. Here's two chromosomes. What's this here in the middle, the circle? That is the centromere. Now, we don't normally see chromosomes like this. With that kind of eye shape, just that long individual strand. And the reason why we don't normally see chromosomes like this is because when they're in this eye shape, when they're the individual strand, when they haven't been duplicated yet, they're loose, unraveled chromatin. They look like this. We don't get to see the individual eyes because they're all unraveled, not very tightly coiled. During S phase, chromosomes duplicate. And they're held together. So this one chromosome became this two chromosomes. Although, and here's the catch, is why we're drawing this. Same thing over here. This guy duplicated into that guy. He copied himself, and they're held together at the centromere. Again, that circle in the middle there, that's the centromere. So it holds them together. And again, here is kind of the confusing part to all this. I don't know why we use the same words over and over again. But these are also called chromosomes. Yeah, there's twice as much DNA here as there is over here, yet we're still just referring to one chromosome. One chromosome, one chromosome, one chromosome, one chromosome. I see two chromosomes on this side. I see only two chromosomes on this side. So in the terminology, well, you can see why we're going over it to begin with. Terminology can certainly get confusing. Okay, we identify these individually as sister chromatids. So this is one chromosome with two sister chromatids. Same thing, one chromosome, two sister chromatids. All right, don't be afraid to refer back to these images. Don't be afraid to refer back to some of these notes, these underlined words as we proceed here, because again, we're going to use these words and they are important. It is important that you understand the difference between a chromatid and a chromatin, chromosomes and chromosomes. As we take a look at mitosis. So there's two parts to M phase. M phase is the division of the cell. The division of cell occurs in two parts. Mitosis is the first part, and it has four phases. So mitosis is the division of the nucleus. The first of those parts is called prophase. So pro means first. Greek or Latin, I can't quite recall, but makes sense. Our first phase, prophase. Try to leave yourself some room. We're going to draw what we see going on over here and make a list of what's going on here. Draw it over here. So first thing that starts to happen that kicks off mitosis, the division of the nucleus, is that the nuclear envelope dissolves. the nucleolus disappears. You guys remember the nucleolus is a very dense protein-filled section of the nucleus, and its job is to produce ribosomes. What kind of dissolves, disappears? The chromatin is going to go ahead and coil up and condense into chromatids. So again, there's those terms we were talking about up above. The centrosomes. Now again, watch the terminology. I wish I could tell you there is a reason. These terminologies are so similar. I don't have a reason for you. All I can say is they are very similar, and so watch the terminology. The centrosomes, not the centromere. We talked about the centromere up here. The centromere was the middle of the piece of DNA. Centrosomes we talked about earlier this week. The centrosomes are a pair of centrioles that make microtubule fibers. They make the asters. So they're going to form what we call the mitotic spindle. The mitotic spindle is those microtubules. Basically, those asters are starting to extend from the centrosome. And finally, if you're at the end of your page, I am as well. Finally, those centrosomes, which again are a pair of centrioles, they start to migrate to opposite sides of the cell. If you remember when we drew them a couple days ago as part of the asters, they were pretty close together. Well, they're going to push off each other and move to opposite poles, opposite sides of the cell. Let's do our best to diagram a cell in prophase. So first thing, I'm going to draw a dissolving nucleus. To do that by using a dotted line, not a solid line, but a dotted line. So there is my dissolving nucleus. I'm going to draw a cell with 4 chromosomes. Is it human? No way. Humans don't have 4 chromosomes. We have 46 chromosomes. I'm not drawing 46 X's. I'm not sure I'd have room to even if I wanted to. So definitely not a human cell. But there's my 4 X's. We're going to split apart here in just a minute. And then finally, let's get this mitotic spindle so you guys have a little better idea of what that is. Again, 2 centrioles. 2 centrioles. A couple days ago, we drew the aster coming out of these centrioles. Well, those are starting to extend, and they're starting to connect with one another, and they're going to start to push those centrosomes, which are made of 2 centrioles, they're going to start to push them towards opposite ends of the cell. All right, first phase complete. That's prophase. So let's move on to the second phase of mitosis. So second phase of mitosis, metaphase. Going to do the same thing. We'll take a list of what's going on. and then diagram it right over here. Oops. So the centromeres reach where they're going. Again, they were pushing off of each other when we last saw. To start metaphase, those confusing myself with my own terminology here. Centrosomes. Wow. And it's not my terminology. I would have named it a lot less confusing if I were the one doing it. Centrosomes, those collection of centrioles reach the opposite ends of the side. Now we're ready to begin metaphase. During metaphase, the microtubules of the spindle fibers. So you can think of that mitotic spindle. Well, those spindle fibers, those microtubules, they attach to the kinetochore. So this word here is kinetochore. So the kinetochore is a part of the chromosome, part of the DNA. It's right in the middle of the centromere. So remember, the centromere is holding two sister chromatids together. The centrosome is the centrioles. They form mitotic spindle. The centromere is the middle of the chromosome, and the very middle of it is the kinetochore. Well, at the kinetochore is a place for the microtubules to kind of latch on and connect. Those spindle fibers, those microtubules, are going to, once they connect to the kinetochore, they're going to start to push and pull chromosomes until they get them in line. So they're going to push and pull those chromosomes until they get them aligned in what we call the metaphase plate, which is basically just kind of an imaginary line. I'll diagram it for you guys here in just a minute. It's at this point. So once we get the chromosomes lined up on the metaphase plate, it's at this point that that third cell cycle checkpoint occurs. If you guys remember the cell cycle checkpoints we talked about, one of them happens between metaphase and anaphase, and that's right here. All right, so here's our cell. We started off by getting our centrosomes to the opposite sides of the cell. Those centrosomes then, they still have their mitotic spindle fibers. I'll draw those in a second, kind of connecting to one another. They're going to latch on and line up the chromosomes. So lining up the chromosomes. Again, how do they do that? those spindle fibers attach to the middle of the chromosome. So both sides, left side here and the right side, are going to grab a hold of the chromosomes. Now that's not all. There are some mitotic spindle fibers that are actually still connected to the other centrioles. And they're going to use that to push off one another here in just a minute. Some that aren't even connected. So these are spindle fibers. These lines we have drawn here, these are the spindle fibers that are kind of coming out and grabbing hold of the chromosomes and lining them up on. You can kind of see it now. This line that we've created is called the metaphase plate. We've got those chromosomes lined up, and the checkpoint, here's what the checkpoint does. At this checkpoint, we have to make sure the spindle fibers are attached to the chromosomes. We have to make sure that the kinetochores are all connected to these spindle fibers. So if, and you don't have to do this on yours, but let's say this spindle fiber missed and it's not connected to the kinetochore. So instead of being connected like it should be, let's say it's down here. Well, with all hope, this checkpoint would catch that and would say, wait, hold up, we got to fix this, and hopefully come over and kind of fix and make sure that they connect. So that's the third cell cycle checkpoint. And if you kind of envision what's about to happen, you can see why that's a really, really important checkpoint. And that ends metaphase, brings us to anaphase. Anaphase, the third part of mitosis, the third phase of mitosis, anaphase. During anaphase, those sister chromatids, they pull in opposite directions. That separates the sister chromatids, it breaks the kinetochore. And the sister chromatids, which have now been split, here's my X, they get pulled apart. Those sister chromatids are pulled to opposite poles of the cell. Here's our cell in anaphase, and we've still got our centrioles. And they're still pushing off one another. So just like these guys were up here, still got some that are pushing off one another. Kind of provides a little extra push, but the chromosomes have broken and are now moving. They're being pulled to opposite sides. Make sure that guy's attached there. So the X's broke apart. The individual sister chromatids are now being pulled in opposite directions. Again, I think we still had one more microtubule kind of connected in the middle there. Let's make sure not forget about that. Those spindle fibers are still pushing off each other. Kind of helps them as they reel those individual chromosomes now back in. They've split the X, and they're pulling those chromosomes, those sister chromatids, to opposite sides of the cell. That way, each new cell, you can see we're going to start to split the cell in half next. Each new cell is going to get an exact copy of the DNA. Okay, our final phase, telophase. Telophase. So prophase, metaphase, anaphase, and finally, telophase. After anaphase, after we have pulled those chromosomes apart, mitotic spindle breaks down. Oops. And so in telophase, we almost have like the opposite of prophase. All those things that happen in prophase are now going to unhappen in telophase. So our mitotic spindle fibers break down, and my whole entire mitotic spindle really breaks down. The nucleus and nucleolus are going to reemerge, the reforming. The chromatids, remember those are the densely packed and tightly coiled versions, they're going to unravel back into chromatin. They're going to go back to their spaghetti shape where we can't really see them. In animal cells, we'll talk about plant cells in just a minute here, but in animal cells, what we call the cleavage furrow forms. So let's go ahead and draw. This one's going to look like a sideways 8 to me, or at least because I'm splitting my cells this way. I guess if I split them top to bottom, it'd look like a regular 8. But I'll keep with my consistency here. So I've got almost kind of like a sideways 8, and this kind of breaks. See how it's kind of going indented right there? You can probably tell what's starting to happen. That break right there, that's called the cleavage furrow. We'll talk more about that on the next page here. So that little indent is our cleavage furrow. And our centrosomes with their centrioles in them, these guys, they kind of go back to being just a regular old aster. The spindle fibers have broken down. Our nucleus is going to reform. So I got the dotted line showing the nucleus coming back. And in fact, we're now going to have 2 nuclei, because we're starting to make 2 new cells. Now, I'm not going to completely draw spaghetti, but I've got my 4 chromosomes that are starting to loosen up. They're starting to go back to chromatin. You can tell they're not as individual as they were up here. And guess what? This guy has the same thing. It's got those 4 chromosomes. Again, they're kind of loose. They're starting to unravel. They're starting to go back to chromatin. We just got a snapshot here, but there we go. We're about to form 2 new cells. And that is the 4th and final stage of mitosis, telophase. That's not the end of M phase. We haven't yet divided our cell. We still have to go through a second part of M phase called cytokinesis. So cytokinesis, all right, that's the final part of M phase. This is the division of the cytoplasm, and it's going to result in two daughter cells. So we just covered mitosis with prophase, metaphase, anaphase, and telophase. That's just the division of the DNA and the nucleus. Now we're going to cut the cell in half. Now I'm going to do kind of two things here, because you got the animal cells and the plant cells, and they're a little bit different depending on whether it's an animal cell or plant cells. We'll start with ones familiar to us. So, whoops, not animal cells, in animal cells, sorry for that. In animal cells, cytokinesis occurs via contractile ring. A contractile ring is like a belt. Man, I'm getting way too close to this side. I need to leave half of this for plant cells. We'll make do, I suppose. So the contractile ring is made up of microtubules. Those microtubules are probably the same microtubules that were a part of the mitotic spindle. So remember during telophase, the mitotic spindle dissolved, it broke apart. Well, we're not just going to waste all those microtubules. Instead, we're going to make them as a part of a contractile ring. So what that belt-like ring is going to do is it just starts to tighten and tighten and tighten until it gets so tight that it eventually just pops the cells into two. Oops. So there's our contractile ring, and it does just as its name suggests. It's going to contract and contract and contract. It forms a ring around the cell until it just gets so tight. So there's our two nuclei. It's just going to get so tight until it eventually pops off into two new cells. each with their own nuclei. And that's the end of cytokinesis in animal cells. Kind of the whole process right there. Just kind of makes that contractile ring and tightens it and tightens it, tightens it and tightens it and tightens it until finally, again, got two new cells. All right, how about plant cells? They're a little bit different. So in plant cells, instead of using contractile ring, they don't have a contractile ring. Instead, there are vesicles that contain cell wall materials. They meet at what was once the metaphase plate. So instead, we've got these vesicles that contain cell wall materials. They meet kind of in the middle of the cell. And with that material, a new cell wall is formed. And that's what creates the two new daughter cells. So a lot less violent than the pinching in half. And draw this animal cell here as a square. There's our two nuclei. There's the cell plate. Again, vesicles are going to be traveling to that, carrying new material, new cell wall material, excuse me, and you're going to form a new cell wall. And voila, instead of violently just pinching in half, like these guys, you just kind of built a wall and created two new cells. So that's the difference between cytokinesis in plant cells and in animal cells. Otherwise, mitosis is really very similar in the two organisms. Okay, very quick. Last little bit of the notes here. We're just about done. I apologize for this being so long. But last thing to discuss, binary fission. So we mentioned at the beginning, we've got prokaryotes, we've got eukaryotes. Prokaryotic division is much simpler, and you'd think it should be. There is no nucleus, there's no centrioles, there's no centrosomes. It should be much simpler. So in prokaryotes, cell division is known as binary fission. Binary fission. Again, there's quite a few differences. We'll talk about more of the differences later. But for right now, here's our bacteria cell. There's its DNA. Again, no nucleus. The circle in the middle is not a nucleus. There is no nucleus in a prokaryote. There's just the DNA. Again, they have just one circular DNA or one circular chromosome, excuse me. And all of cell division for them is basically the DNA replicates. So we had one circular chromosome, now we have two. And then you have cytokinesis. And you end up with two new cells. How about that? Much, much simpler. Quick note on all that. I'm not saying it always does, but in bacteria and prokaryotes, it can take as little as 20 minutes versus about 24 hours for eukaryotes. Whew. All right. What a day of notes. I need to drink water. My voice is, well, I'm losing it. So I'm going to call it quits there, and I'll see you on the flip side. Have a good